Robert Doerning. I am from Cold Bay. I worked in the Ickert Bond Lab working on distinguishing Claytonia scammaniana from the other Claytonia species. Claytonia scammaniana is also known as spring beauty. Its petals can be white with pink or purple veins, as you can see here. And it's slightly yellow, if you can notice, because it's been pressed under a herbarium sheet for many years, some even going back 100 years. Basal leaves are strictly linear with rounded tips, as you can see right here. And it has 2 sepals, 5 petals, and 5 stamen. The sepals are the green leaf-like objects surrounding the base of the petals. The stamen are the male reproductive organ, and the petals are, of course, the pretty part everyone likes to see. Claytonia scammaniana grows throughout Alaska and is commonly mistaken among botanists as Claytonia sarmentosa and Claytonia arctica sensiporcil due to similarities in phenotypes. Now, phenotypes, they are the characteristics, like what you look like. For example, in people, your eye color, your hair, how tall you are, all that, that's what you inherit, and that's what the phenotype is. And as a side note, Arctica sensiporcil means Arctica according to Porcilled. The work in this lab utilizes information from O'Quinn and Hufford's review of the Montier subfamily. Using DNA sequence data, Hufford and O'Quinn improved our understanding of the molecular systematics of species within the Claytonia and Montier genera. And what I have here is the little graph I made, kind of proud of it. Montier is the subfamily, Claytonia and Montier are the genera, and those question marks are species. And what they were doing was finding out the relationships between these two and the species. The primary goal of this project was to undergo a more in-depth look into Hufford and O'Quinn's study. By using their methods, Claytonia scammaniana will either be identified as a distinct species of Claytonia, identified as the same as another species of Claytonia, such as sarmentosa or arctica, or it will be unidentifiable. And that will be because of a mess up in procedure. It could be corrupted DNA, and then lack of data. It could be any one of those and more. And another interest is Racematosae, and this is an interest because it is well represented in Alaska, and our samples are from Alaska. The sample used in this project is C. scammaniana, Claytonia sarmentosa, and Claytonia arctica sensiporcil. The DNA extraction was used on each sample by using a QIAGEN D&EZ kit. And then they were put through PCR and the thermocycler. Now, PCR, which is polymerase chain reaction, is a process made of 3 steps, repeated around 34 times. And what happens is you start off with denaturation, which is done around 94 degrees Celsius. And what happens there is the single DNA gets split into 2, so you have 2 single-strand DNAs. Then you move on to what is called annealment. This is done around in the 52 degree Celsius range. And what happens is DNA primers attach to these DNA strands. After that is around 72 degrees Celsius, and that is elongation, which is where Taq polymerase attaches to these DNA primers and then use that to elongate into new DNA. So you have 2 new DNAs from one. And this is happening, again, around 34 times with millions and millions of pieces of DNA. We then use electrophoresis to separate our PCA amplicons with gels. And right here we have our ladder, which is like our benchmark for our concentration. It's what we refer to. And right there is what we're looking at, and that's about 1 kb, which is 1,000 base pairs. A successful one would be right here, and that is about 700 base pairs, which will be used for further analysis. And we got that with multiple gels. PCR amplicons were purified using a PCR purification microfusion vacuum protocol made by QIAGEN. The purified PCR products were sequenced using an API 3130XL genetic analyzer residing in Institute of Arctic Biology DNA Core Lab, which is in Fairbanks, UAF. The sequenced DNA aligned The sequenced DNA was aligned using a sequencer software, and data from O'Quinn and Huffer's work was included. And when we use the sequencer software, what it does is set up a large table with thousands of base pairs, base letters, which is your A's, T's, G's, and C's. And what those are, they are the blueprints for life. They make up our information, they are coding. And we arrange them so that we can compare the different base pairs so that we can see the relations. And we use that information to create our phylogenetic trees using RAHI software on the Cypress portal. My results. This table is my cladogram tree, and we're focusing on this area, which is blown up to right here. And any non-showing bootstraps is considered irrelevant in the study. If it's not supported, we did not show it. And we found that section rhizomatosae is supported as a monophyletic, but the relationships within that are largely unsupported. And as a byproduct of this, we also found that the putatively new species Claytonia noaticensis is tentatively supported as a sister to Claytonia sarmentosa. This phylogram tree, again, any non-showing bootstraps are considered irrelevant, they're unsupported. But we found that Claytonia scammaniana is supported to be part of clade with Claytonia sarmentosa and Claytonia noaticensis. But support for relationships within that clade is lacking. We don't have enough. Our conclusion is that relationships within the section rhizomatosae are largely unresolved, and there's just not enough data to support that further relationships within the clade. Also, we found that Claytonia noaticensis is related to sarmentosa, which is pretty interesting. And although Claytonia scammaniana could not be distinguished as its own species or even for sure related to another, we did accumulate data that will help us make our informed decisions in further studies. There's just not enough data to make a finalist conclusion, and that is common in science. By continuing this research and distinguishing Claytonia scammaniana from other Claytonia species, a long-enduring mystery can be solved. It's so often confused with sarmentosa and arctica, and further studies will solve that problem. I'd like to give my thanks to RAHI and the RAHI director, Denise Huertas, University of Alaska Fairbanks, Idea Network for Biomedical Research Excellence, Biomedical Partnership for the Research and Educational Pipeline, National Institutes of Health, my PI who could not be here, Dr. Steffi Kerban, my lab mentors, Stephanie Jeffers, Jordan Metzgar helped us a lot, and then Monty Garut for any help he gave us. I'd like to thank my teachers, Drs. Kathy Katranis and Dr. Adrian Orr, and my lab partner Rena. She was like my third teacher, I guess, and then the RAHI TCs who helped me along. Thank you. And Richard, because he's super smart.